Okay, hi, welcome everyone. On this beautiful spring day to the second class of Introduction to Systems Biology. And welcome also to those looking at us through the video on your computer screen. Hello. Welcome physicists, welcome biologists, welcome computer scientists, welcome engineers, welcome others. We, as always, start off by an invitation to take together a nice deep sigh of relief like this. In this class, we're going to study about our first gene circuit that goes beyond the simple gene regulation step. And I'd like to start with a review of something we did last time, then pose the problem of how can we speed up responses, and then look at this circuit together. The goal is to understand it intuitively and mathematically. So, our goal is to show how negative autoregulation can speed response times. I'm going to explain the terms. So, last time we talked about the cell, the cognitive problem of the cell, to receive signals from the outside and change its composition accordingly by making the right kind of proteins, the right time, and how this computation is carried out by networks of regulation. We talked about transcriptional networks where proteins called transcription factors receive signals, click into an active conformation, go to the DNA on the time scale of seconds, and increase or decrease the rate of production of a protein encoded in the genes that they regulate. And we analyzed the dynamics of simple gene regulation last time, where we had a concentration of protein Y, which is produced and removed. Its production rate and removal process. And we saw that the removal is a sum of the rate that proteins are chopped up by the cell actively in an energy-consuming process, destroyed, and dilution, which results from the fact that cells are growing. The cells are growing exponentially. The cell grows to twice its size and then divides. And so if we have a certain amount of proteins in the cell, suppose there's no production and no active degradation, just the fact that the cell grew by a factor of 2 means that the concentration goes down continuously. Every cell generation time goes down by a factor of 2. So that initial population gets diluted out exponentially by the increasing volume of the cells. And it's related. This is the generation time. Time in which one cell doubles itself, which could be, as we said, 30 minutes to a few hours for E. coli, and it could be a day for cells in our body that are dividing. And we kind of solved this equation in the situation where there's where we start with no proteins, and then and produce them at a constant rate. And we saw that the resulting dynamics, they reach a steady state, which is the ratio of production and degradation rates. I'm reviewing what we did last time. So the more faster you produce, the higher the steady state. The faster you remove, the lower the steady state. And you start producing them. So this beta controls this rate here. And then when you have more and more proteins, the removal term becomes appreciative, and you end up like this. And we defined something that will be a topic here, the response time, which is in any process, right? This is time to reach halfway from initial to final levels. The steady state level, in many cases, the level of the protein made is important. It shouldn't be too high or too low. There's an optimal level for the functioning of that protein. So the steady state is important. If we want a circuit that makes, let's say, 1,000 proteins per cell, the next question is how fast you reach there. How fast you reach, let's say, 50% of that. That's a typical time scale that we're interested in, just like in engineering. A lot of times, the faster, the better. So this response time, T1 half. And you remember, we calculated it for this system. And we noticed that the solution to this equation is y of t is a steady state 1. I mean, this function, which starts linearly and then kind of converges to the steady state. And we asked, so at T1 half, this response time, we reach halfway to the steady state, right? This is T1 half. This is halfway to the steady state. And to calculate that, see, this falls out. And the answer is depends only on the removal rate. It doesn't depend at all on the production rates. This might seem surprising, right? If we produce more, we make more proteins quickly. So why is it that this halfway point doesn't depend on the production rate? So let's imagine that we increase the production rate. Okay. So what will happen if we increase the production rate? We'll reach a higher steady state, and we'll also produce more quickly. But the time to reach halfway of that new steady state will be exactly the same. Yeah.
But if we need a certain concentration to respond, that we will get to that concentration quicker.
That's right. The question is, if we need a certain concentration to respond, let's say 100 proteins, increasing the production rate, you'll get there faster. The reason response time is useful is because the steady state concentration usually is in the range of the functional amount. So that is the amount. So half of that is like the amount you need to respond. So how fast you reach the amount you need to respond is connected also to the steady state level. But your question is quite important, and actually we're going to use it for speed up by decoupling the production and the steady state.
But if we're recording the half time, of course it's not
Yeah, so let's question about increasing alpha. So suppose we want to speed the response time by increasing alpha.
It's the thing, if you want to run faster, decrease the length you need to run.
Yeah. This is all very important questions to understand the constraints here. For example, let's analyze what happens if we. So one strategy to speed up. By the way, is this writing big enough for the camera? To speed up, one way we can do is remove proteins faster. Let's increase alpha. For example, start breaking up the proteins actively. So this will cause the steady state to go down. But we want a certain steady state. We want a certain functional amount of that protein. So we need to increase production in order to balance out. But to keep the same steady state, we then need to make more proteins by increasing beta. And this has a cost. So we can make the system faster to reach the same steady state by increasing alpha. But there's a cost. There's a fetal cycle of making and breaking, 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 making, breaking, making, breaking. And since organisms like E. coli are limited in the amount of proteins they can make, it's a waste. So they actually don't use this strategy very often. Most of their proteins aren't actively degraded on the timescale of a cell generation. So the question is, how can we speed up the responses given a certain steady state without needing to make and break proteins? Right, so that's our question. One moment. How can we speed up T1 half without making and breaking unneeded proteins? By the way, these equations, as simple as they are translated to experimental predictions. And you can do the experiment, for example, just to give you a feeling for the technology today. You can take E. coli, like I mentioned last time, take this green fluorescent protein from jellyfish, this piece of DNA, and connect it to a regulatory region, a promoter, and you can write in the promoter whatever you want, the binding sites for transcription factors, etc. Play with the production rate beta by making mutations in the DNA that to which bind the proteins that make RNA. So these mutations change the chemical affinity of the machinery of transcription to the regulatory region. And you can change those letters experimentally to play with beta as much as you want. You can play with alpha by, for example, making that protein that you make less stable, more stable, degraded more often. You can play with those numbers experimentally and measure these dynamics because the cells now become fluorescent, green fluorescent bacteria. And with a fluorimeter, you can measure this very accurately. And did you see, for example, that the steady state depends linearly on the production rate and inversely on the steady state, and that the response time is one cell generation, the protein is stable. One reference for that. So these equations work inside the simple context where they apply. They also work for human cells, etc. The power, the experimental power is awesome now in biology to read and write into the DNA and build new circuits. Yeah.
If I want my protein to both respond quickly but also increase its steady state level, wouldn't I have a problem?
So what if I want to increase the steady state level and make it respond quickly? good question. I'm not going to answer it, but you have the tools to answer it after this class. I hope so. So to answer this, we're going to actually look at the actual how the transcription network looks like and learn from E. coli the circuit that does this. So I'm going to introduce to you the concept of network motifs, which are patterns that appear in the transcription network of that we're looking at much more often than you might expect at random. So to remind you, we talked about transcription networks where protein X changes the production rate of protein Y, which can then change the production rate of Z and W, U, right? And when you look inside the E. coli and all organisms, R cells, etc., you see transcription networks of this kind. The signs on the arrows can be positive or negative, as I said last time. Now let's look at this as a graph, which has nodes and edges, points and arrows. And look for patterns, it can be small patterns, subgraphs, that occur in this graph again and again and again. And then we can ask, does it make sense that they occur in these numbers by comparing their number of occurrences to randomized graphs that have the same properties as the real graph? And in this way, zero in on properties of the graph which are unexpected. That natural selection, in fact, needs to keep selecting these circuits in order to have them, to avoid having them washed out by mutations that occur. And we're going to talk about this for this class, the next class, but we're going to talk about one particular kind of, you can do this systematically for all small patterns, etc., which we'll talk about. But we're going to look today at the pattern, the simplest pattern, which is a transcription factor that regulates its own production. That's to say, in biological terms, this is the DNA. This is the gene that causes the production of Y. This Y binds a site, it recognizes chemically a site in the promoter of its own gene and changes its own production rate. This is called autoregulation. Just to play, we're going to look at a part of the E. coli network that has 400 nodes, that's to say dots, and 500 arrows. That was well studied for this particular purpose. It's a part of the network that's really well worked out by many different labs and experimental techniques. So we'll have 400 nodes and 500 arrows. Now in this network, in the real E. coli network, 40% of the transcription factors have autoregulation. They're mostly auto-repression, or what we'll call here negative autoregulation. Negative autoregulation. That means Y represses its own production. See? That's 40%. Is this, in a network of this size and density of arrows, is this number you might expect for any network of this density and size, or is this unusual? So we can calculate, we can compare to random networks. What is the expected number of these self edges.
Don't really expect a random network. Because in a random network, things will take so long that E. coli will die before it completes a cycle. Because you start with one protein, move to another, then to another. Random walks will be far longer.
Yeah. So the question, the comment is that inside E. coli we don't expect a random network, and that's exactly the point. The networks in the living cell are the products of natural selection, of evolution. So they're not random. And we're going to find out soon that this pattern, the number of times it occurs in the real network is much, much more often than the ensemble of random networks. And the reason is that we'll argue is because it carries out a function. So this random network. Why are we comparing to random networks? It's a useful way to zoom in on the patterns that are unusual, and we claim that they're unusual because they're in a living object.
But the question, which of the constraints actually lead you to them? Because if I would just say, let's limit random walks to be no longer than 3, and then look at the graph and it's still overexpressed versus a random graph, then you say that there's something more to it than just the need to complete things.
I mean, once we find a pattern, we'll have to ask, why is it there? And there we can do theory and experiments, and this is a different question in each case, etc. By the way, if we take an organism like E. coli and put it in a situation where conditions are constant, that is to say, it doesn't need to change its composition anymore, it doesn't need to think anymore. You can say it's in a constant environment. And this actually happens in nature almost. There's relatives of E. coli that have become addicted to living inside cells or inside termites, things like that, where they're supplied with constant conditions. Over evolutionary time, they lose all these arrows. And the actual null model is a network with no regulation. They just make everything at a constant rate, optimal rate. So each arrow here actually has to survive, is there for a reason. To remind you, if I make a mutation, a copy of the DNA, and there's an error in one of these 10 letters here in the binding site, that weakens the interaction by about 10-fold. So with 1 or 2 letter changes, I get rid of an arrow. These 1 or 2 letter changes happen very often. If I take a cup of water with some salt and pepper and I grow 1 E. coli there to 10 to the 10 E. coli's, there's 10 to the 10 duplications of that original DNA. The error rate is 10 to the minus 9. I'll have 10 examples of every single base mutation in that cup. So after, not so many generations, I sampled all the mutations. If one of them is growing faster than the others, it'll take over. So there's very quickly you can lose arrows if they're not needed. You can make new arrows by mutations that make a binding site or a piece of DNA that jump and bring a binding site from one. So these networks on the timescale of, let's say, hundreds of generations are very plastic. They learn quickly. And therefore, what's there is there for a reason. So just to do this calculation, we have 500, let's say 400 nodes. And I'm sorry. Just looking at a random network, 400 nodes, just for example. 500 arrows called edges. And if we imagine just the simplest kind of random network, you can do much more sophisticated things, where I keep these 400 nodes and I take 500 arrows and throw them at random. And I generate a random network. And then I look how many times is it that an arrow can begin in any node and end at any node. I don't make any other constraints. So sometimes an arrow will start at a node and end up at the same node. And I do that, and I count how many times that happens. And then I make another random network, and I count how many times I get an arrow that is self-regulating, and again and again and again. And I average over that. I do the statistics. So the chance for self-arrow, let's analyze it. I start from a node, and I can hit any one of the other n nodes. So what's the probability to hit myself? P self is 1 over the number of nodes. And I do this E times. So the mean number of self edges n self is E times the probability to have a self edge, which is 500 over 400. It's about 1.2. So at random, I expect about 1. What's the standard error, standard deviation in this process? This is a little bit like a coin tossing. I toss a coin, there's a probability of 1 over n to fall on its head. And I do that 500 times. The standard deviation here is similar to a Poisson process. So standard deviation is square root of the average, about 1 again. So I expect 1 plus minus 1 self edges. But in the real network, I wrote percent, but actually what I meant is there's 40. It's not 40%, there's 40. I made a mistake there, sorry. 40 transcription factors with autoregulation in the real network, which is, as you see, more than 30 standard deviations away from what you expect. That's hugely unlikely. Of course, comparing to random networks, which this is one way to think about statistics here. So these are this negative autoregulation is a network motif. It's an interesting pattern that occurs again and again and again. It happens also in other organisms, not only in E. coli, including our cells, again and again and again. So we can ask, what is the advantage it gives to the organism that might lead to the fact that this arrow is not eliminated by the mutations that occur naturally? So we need to now understand the function of this. Good. In the next lectures, we'll look at other network motifs, more elaborate circuits that occur here. We can learn interesting things from them. One moment. So we review network motifs, autoregulation, negative autoregulation. Before the question, it's a good time, I think, to take another nice deep sigh of relief. Yeah.
But something about the you code for a protein, and then the code produces a protein and has a larger affinity to connect to the place that is coding the protein. So it's some sort of a circular.
So the question is, you produce a protein and it connects to the place that produces the protein. So I'd like to explain myself a little more clearly. The place that produces the protein we call the gene, and the thing that place that regulates the promoter, the gene is called a promoter, and it's not the same piece of DNA. The location is different. Sometimes these sites can be inside what's called the coding region. That can also occur, and then it's evolution has more constraints there. Absolutely. Good question. And these questions like this are very important. If you don't understand something like this, I bet you that other people will really benefit if you ask. So. Yeah?
What is the disadvantage of this autoregulation? Will disappear if everything would be just flat? And why won't it stay and just be there?
Why won't this stay and be there? That's a question?
You say that basically the evolution will cause those autoregulations to disappear whenever they are not
Needed.
needed. Why?
Why would it make them disappear? If there's a mutation and that bacterium is growing faster than the other bacteria without a mutation, it's an exponential growth process, and that mutation will start becoming more and more common and eventually take over the population. So if a certain property like a negative autoregulation is no longer useful. In fact, has disadvantages, or neutrally just equal advantage, it'll tend to disappear.
So why is it so disadvantaged? What's the problem about it?
So actually, it appears more often than you might expect. So the question that we ask, what is its advantage? Maybe I didn't understand. What's the disadvantage about? What's the reason that it increases the fitness of the organism? Oh, okay. Maybe try to ask. All right. So our task is now to understand what negative autoregulation can do. And so let's think a little bit about negative autoregulation. Negative autoregulation, NAR. So again, biologically, this gene codes for protein Y. Protein Y is made to recognize a site, that is to say a certain sequence of letters, bind here, and by that decrease the rate at which there's transcription of that gene and therefore production of Y. So the equations that govern this particular situation, the rate of production of protein Y, there's a difference from simple regulation, and that is that the production rate is now a decreasing function of the concentration of Y, right? And we're soon going to prove something about the dynamics of Y, no matter what the shape of this decreasing function. We're going to show something that only matters that it's a decreasing function. But I'd like to still calculate for you one example of this decreasing function that occurs very often in biology. This shape, in fact, for most regulatory reaction, not only negative autoregulation, but most repressors, X represses Y, have a certain family of shapes here. And these family of shapes is very useful for modeling biological systems, is usually these regulatory functions have a shape called Hill function. And that shape looks like this. And draw it. Looks like this. It has 3 parameters: the maximum production rate, the halfway point K, and the steepness is governed by this number n, which is called the Hill coefficient. I was just wondering, how many people here have seen Hill function before? It's about 10%. I'm going to derive soon where it comes from molecularly, what these numbers mean. I'm going to do a little bit of math just because it's important for you to understand where K comes from, because we're going to use it in a while. But I want to say that this n governs how steep the function is. So if n equals 1, it looks like this. And if n equals 4, it looks like this. If n is very large, it looks like a step function. And in biology, usually the Hill coefficient ranges between 1 and 4. It comes from the fact that these repressors work sometimes as dimers, tetramers, 6 proteins together in a complex, this multimolecular reaction. This K is the concentration of Y needed for 50% repression. It comes from the chemical bonds between Y and its DNA site. So if I make this site really perfect for this protein, I make these, let's say, 10 letters exactly match the shape of Y that recognizes DNA, this K will be small, and it'll need just a little bit of Y to repress. And if I start making a change, like I said, a one-letter change might change K by a factor of 10. Now it takes 10 times more Y to repress. If I make two changes, factor of 100, and maybe that's not relevant anymore. Y never represses, and I lost the arrow. This number, I just want to stress, this number is hardwired, and it's the same from cell to cell if there's no mutations. And most cells, by the way, don't have mutations. It's rare to have a mutation. Right? So in a population of cells, this number is very constant. It has to do with the sequence of the DNA, which is the same, the structure of the protein, which is the same. But this number varies by tens of percents, let's say 30%, from cell to cell. And this is something I talked about a little bit. The biological material is noisy. Because transcription and translation go through bottlenecks of small molecules, small numbers of RNAs, there's stochastic noise in this. And also things about the metabolic state of the cell, etc. So when you look at the level, the beta over alpha, the steady state level of a protein, in a population of cells which are in the same conditions and have the same DNA, etc., you'll find that neighboring cells have 30%, like a distribution where the standard deviation is maybe 30% of the mean. So that's a problem also. Okay, so I just want to say that production, beta M, is noisy, which means, let's say, typically 30% cell-to-cell variation in this. So this doesn't change from cell to cell. This changes from cell to cell. Steepness also doesn't change from cell to cell. So these are the properties of regulation functions. I just want to say that for an activator, this is a repressor. For an activator, beta is, of course, an increasing function. And the Hill form is very similar. It just has this y over k to the power n up here. So this is the Hill functions. Mm-hmm. What? This is for an sorry, thank you. This is for an activator where Y increases transcription, production of protein. This is repressor where Y represses. I just want to tell that what we're doing here also depends a little bit on separation of timescales. This Y here binds and unbinds the DNA with a time scale of, let's say, seconds. And in the case of a negative autoregulation, Y affects its own production rate. But the level of Y changes much more slowly. It's governed by this half times, alpha, which is like the cell generation time. It's 30 minutes or something. So what we can do is, at every particular moment, look at the concentration of Y, and imagine that it's constant over the 1 second time scale where we compute this production rate. And therefore, we can separate timescales and use a static function, like beta is just a function of the concentration of Y at that particular time. And on slow time schemes, Y will change, then change this beta of Y. That way, we can understand the system quite more simply. If all these timescales were the same, it would be much more complicated to understand the system. Yeah. All right, so I think this is a perfect time to take a break. I'll take a 10-minute break, and we can end by just taking another deep sigh of relief. And I hope you enjoy the break and come back. So, welcome back from the break. As people are sitting down, let's start by taking a nice deep sigh of relief. Which, by the way, is a great thing to do when you're working in your computer, and it tends to be, it's very relaxing, puts you in the moment. And so you can do it also outside of class if you feel like it. All right, so we talked about network motifs. We saw negative autoregulation is a very recurring pattern, and we want to understand some of the things it might do. And we saw negative autoregulation, production of Y decreases its own production rate. And I'd like to talk about how this can speed up the response time without the need to make new proteins, to degrade proteins, etc. And I want to show this to you in a way that doesn't need, we don't need to care exactly what's this function. So I wanted to look at simulation again in a little bit a new way. So here beta is, and here's how we're going to analyze it. Here I'm going to write the concentration of Y, and here I'm going to write I'm going to draw the production and the removal. So the production doesn't, in simple regulation, doesn't care about how much Y there is. It's just beta. It looks like this. Removal goes up linearly with the concentration of protein. Looks like this. The rate of change of Y is the production minus removal. So this here is the rate of change. It's the distance between these two lines. Now, when production equals removal, the rate of change is 0. That's what we call the steady state. Where is this point? At the intersection. That's right. If we have less Y than the Y steady state on this side here, production is more than removal. So the rate of change is positive. We're moving this direction. Y increases. If we're here, removal is greater than production on this side of the steady state. And therefore, rate of change is negative, and we're moving here. No matter where we start, we come to the steady state. Therefore, we can say the steady state is a stable fixed point. It's the only stable fixed point. How fast do we move towards this fixed point? The rate that we move towards this fixed point is the rate of change. The larger the distance, the faster the change, right? So suppose I start here at 0. This difference is quite large. I start moving to the steady state quickly. But as I approach the steady state, this difference becomes small. So I start slowing down. Did I explain myself? So if I draw Y as a function of time, I start quickly and I slow down as I approach Y steady state. Now, this is simple regulation. Now let's compare it to negative autoregulation. And I'm going to make the comparison in a special way that's called a mathematically controlled comparison. That's to say, I'm going to make a fair comparison. This was coined by Michael Savageau in the 70s. I'm going to make this comparison as fair as possible. So I'm going to keep everything that I can about the two circuits the same. So what do I need to keep the same? Same one, steady state. So I'm going to make negative autoregulation and simple regulation to have the same steady state. What else can I keep the same? Same alpha. That's to say, they both remove proteins at the same rate. Because we saw that if we remove proteins more quickly, we need to produce it more quickly, and that gives a cost. So we keep these two things the same. Right? So I'm going to need to draw here the same situation for negative autoregulation. Now, we know that negative autoregulation, this beta, the production, isn't constant. It depends on y. How does it depend on y? It's a decreasing function because it's a negative autoregulation. Y decreases its own production. But they have to it has to cross. But the removal is the same because alpha is the same. So this is the same in NAR and in simple. So this line here is the same. But the production needs to cross at this point to keep the same steady state. And it's a decreasing function. What does that mean? It has to start here high. If it starts here, it can't cross. It has to start high. So it starts here, at beta max, and crosses here. Somehow. So it reaches the steady state. Again, it's always production is higher than removal here. Removal is higher than production here. So it's a single fixed point, one steady global. Globally, there's one stable fixed point. What about the speed? At every point, this blue curve, negative autoregulation is farther from removal than simple. That means that at every point here, the rate of change is larger. That means that at every point, the rate of change is larger. You go faster, and you reach the same steady state. Faster. Hold on. The same principle when I come, this is when I came from 0, when I come from above. If I compare this distance to this distance, the rate of change is larger for negative autoregulation. So if I came from above, it will also be faster. No matter what the shape, as long as it's a decreasing function. Did I explain myself?
So it means that every gene that is regulated in this manner has to start from a very high concentration. What you're saying is basically it's easier to fall from a mountain than to climb it. But
to say the gene has to start from a high concentration. In fact, I want to sharpen up. It has to start from a fast production rate. That's different from fast. So if we use a car analogy, you can drive fast if you have good brakes. What you do here is you build a gene that if it doesn't repress itself, it has a fast production rate. You do that by making mutations that make sure the RNA polymerase, the machinery that transcribes, has high affinity to that promoter and makes a lot of RNA. So you have high beta M. And at the same time, you make sure that this Y represses itself. So you start pressing on the gas quickly, and then you hit the brakes.
But do you pay a price for driving so fast until you hit the brake?
Do you pay a price for driving so fast?
You would pay more fuel for driving very quickly.
Yes, you pay more fuel. Here you reach your desired amount of proteins faster, and it's not like in the car in that respect. There's less of a cost. Is there a limit to how fast you can be? Of course. You see, you can't make this beta M infinitely large. It's limited, for example, by the maximal possible promoter activity, which is about 1 RNA per second. That's used in E. coli, for example, for things like ribosomes, where it needs hundreds of thousands of copies per cell. So there's a limit to how fast you can be. And, of course, I neglected here the delays, like the 1 second binding time and the 1 or 2 minute time it takes to make the RNA, and from the RNA the protein. I neglected that here. So there's a delay before you start seeing anything. So you can't override that in this way. Those delays I neglected here because we're doing a theory that has to do with the 30-minute timescale, hour timescale. And again, you can see this experimentally. The same paper I mentioned, Rosenfeld, actually an experiment done in my lab, took the gene of a particular repressor and connected it to green fluorescent protein. So now you have a protein that both can bind to DNA and is green fluorescent, and made 2 different bacteria. Here, he programmed the binding site that this protein can recognize. And growing the bacteria and looking in the fluorimeter at the fluorescence, you get curves that are very different steady state, because this represses itself, it reaches a lower steady state. But when you normalize to the same steady state, you see that the negative autoregulation speeds, in this case, by 5-fold. So instead of, let's say, a 1-hour delay, you have 12-minute delay. And this was also seen in the last decade in several naturally occurring systems which have negative autoregulation, like DNA repair in bacteria and some systems in some receptors in the immune system, etc. All right, so negative autoregulation speeds the response time. That's one thing it does. I want to look a little bit more detail at how the properties of this speed up. And for that purpose, we're going to use the actual forms of repression functions. And so remember, and these can look quite sharp, and they have a kind of threshold, K, where Y is less than this concentration that's determined, again, by the chemical bonds of Y to the DNA. Then you have high production, and when K exceeds this concentration, you have low production. It's quite a sharp-looking function. So what happens when this function is quite sharp-looking, I want to ask? It has another nice thing that happens. And now, first thing you notice that if it's a sharp-looking function, then the steady state is very close to this threshold k. Because this function starts somewhere high and then goes low. And the place where it crosses this line is, in the limit of a step function, it's exactly k. No matter even what the removal rate is, I can play with the removal rate. As long as it's not too high. They always cross here at the threshold. You see that? Even if the removal rate was a little bit higher, so k. I can make this beta m large. Doesn't affect the steady state. I can play with the removal rate. Doesn't affect the steady state. Making beta m large affects the speed up. I can make the speed up really large if I make beta m much larger than this beta here. This beta, by the way, for simple regulation, you kind of drive slowly to reach the desired steady state. But if I make beta very large, I can drive quickly and then stop at this k. So what's going on here? With negative autoregulation, you can program by mutations this value k. It's basically the ratio between the off rate and the on rate of the protein to the DNA. And that determines where your steady state will be. With another set of mutations, you can program how much this gene is transcribed when there's no Y. That's another piece of the DNA. Let's see here. This affects beta. Yeah. So you can decouple those two. You can decouple the steady state from the production rate. Yeah?
Can you please repeat why k is equal to the y constant?
Yeah. I will. I'm going to deal now with sharp, step-like, almost step-like regulation. So it looks like beta m, and then it goes to 0 at k. I want to make sure I understand your question. Your question is why the steady state is equal to k?
Yeah.
Thanks. So the steady state is when production equals removal. And the property of step functions is that the straight line can only cross, if the straight line has this slope here, it can cross here. But for a large range of removal rates, it doesn't even matter so much. It always crosses here at the threshold, at the place where this function is changing very steeply. Yeah? Or I'll explain to you another way. Suppose I start with zero protein. Zero protein, so I'm below the threshold, so I start making it. When I reach this point k, I stop making the protein. So you make it, and then when you reach this point, you stop making it. So if there's any delays in the system, it's very important to say, if there's any delays in the system, you might get a little bit of oscillations. Because you go above the threshold, now stop producing, so protein decays down. Now I'm below the threshold, so I start producing. These are usually damped oscillations. I like to give the metaphor of the shower that I had when I was growing up in Kfar Saba, which I tried to fix the water temperature, and I would make it too hot, but it was only a delay. So and then I cool it down, then it was too cold. This caused some oscillations. So this can happen if there's delays. Settle down at this point where you change from high production to low production. Did I explain myself? Good. So the point is, makes Y steady state insensitive to variations in production, like beta max, and removal alpha, it locks it at k, which is hardwired, same for all cells. What does this do to us? It solves another problem that we have, this problem of biological noise I talked about. Not only does the circuit make you reach the steady state faster, it also stabilizes the steady state against the most fluctuating elements in biological material. This variations in beta that I mentioned. And this is a principle we'll talk about quite a bit of robustness. It makes the steady state robust with respect to the most variable or noisy parameters. In this case, it's beta. But also, the removal rate, let's say if the cells are growing faster or slower a little bit, doesn't change the steady state.
K is the infinity?
What?
K would be the infinity?
Yeah, K is the affinity. Yes, yeah. I'm tempted to show you how all these functions come from basically statistical mechanics of sticky billiard balls binding to a site. That's all. It's just a partition function. It's just statistical mechanics. K is has to do with the exponential of the free energy of binding, like the Boltzmann factor, basically. In contrast, simple regulation, Y steady state is sensitive to changes in beta and alpha. Y steady state is actually equal to beta over alpha. So let's say a 30% change in beta would lead to a 30% change in steady state. But in the case of negative autoregulation, a 30% change in beta wouldn't do anything to the steady state in case of a sharp function, which you often see. Yeah?
How do you control, how do you biologically achieve this step-like function?
So biologically, the step-like functions result from the fact that these transcription factor proteins Y that I drew like this are actually almost always multimers, like dimers, tetramers, hexamers. And so you need, to put it most simply, let's say 4 of them at the same place at the same time. It's not exactly like that, and that leads to concentrations of power 4. So it's by these making or to have them bind at 2 different sites where they help each other bind. So one binds, and then that helps the other one bind. And this is like a cooperative process. Cooperativity is what leads to sharp input functions. Again, this is a topic that, I mean, how many people here heard about cooperativity in biology? And this probably identifies the biologists in the audience. It's very common to have these steep functions. And you can make models, again, statistical mechanic models for multiple binding events and derive this function.
Explain again how come changes in alpha won't matter in case of a step function? Because it seems that let's say alpha is steep.
This is the steep function. And now if I change alpha, let's say I increase it a little bit, change the slope like this.
It seems like
This is the crossing point, steady state, where they're equal, production and degradation is equal. Because of the steepness, it's almost the same as this, or it's the same if it's infinitely steep.
K unified with Y steady state.
Excuse me?
Assuming that K unified in Y steady state in the theoretical step function.
If it's a pure step function, Y is equal to Y steady state. You can imagine that if it's slightly curved here, it'll move a little bit. And then in the exercise, you'll solve for a given Hill coefficient n. How much does a certain percent change in alpha lead to a percent change in Y steady state? And the answer is something like 1 over n. So infinite n means the perfect step function. So this is you see, in the case of simple regulation, let's just do it graphically. If I increase, I move the steady state. Or if I change alpha, I move the steady state. But in the case, it moves much less if it's a sharp function. Okay? So,
Question?
one moment. Negative autoregulation. Speeds, response time, and makes steady state robust with respect to production removal. Yeah?
Alpha could depend on y also?
Okay, the question is, can removal rate depend on y? And the answer is yes. You can have y that there are proteins that increase the rate of their own degradation, for example, etc. And we'll talk about cases like that in the morphogen, in the how to get from an egg to an embryo. This work can be quite important. And then I think with this tool, I hope you can now analyze it. If y is not a straight line, but an increasing line or decreasing line, I invite you to think about these questions like this. You can actually prove for whole classes of functions statements like speed up, and which you'll do in the exercise. For instance, you'll show that if y increases its own production rate, that slows down the dynamics. And that actually happens in 10% of equalized transcription factors. They positively regulate themselves. And then you can ask, when is that useful? In what situations could that be useful? Also, it increases the variability between cells, positive autoregulation, the opposite. So I want to just suggest to you that sometimes living organisms are quite different from engineered devices in that they use noise. Like I pressed the button on my radio, I want it to work 100% of the time. Otherwise, broken radio. But if you take a living organism, one of the things that defines it is that it doesn't do the same thing all the time. Right? Otherwise, we say it's robotic or that occurs in all bacteria and humans, etc. And so some cells use the random noise, random number generator, to make different behaviors. Also, we'll talk about that later in the courses. I'm going to finish with this story. We have two organisms. They're identical. One moment. Organism A and B are identical, except that organism A has simple regulation on gene Y, and organism B has negative autoregulation on gene Y, made to reach the same steady state. In a situation where you want fast response times are given advantage, we send them out to nature. Organism B makes protein Y faster and in a more stable way, in a more less variable way. It therefore makes more babies. Those babies survive more. They make more babies. They survive more. Over time, negative autoregulation is what you see. And that is how natural selection can produce things like circuits, like negative autoregulation, which maybe an engineer would learn in engineering school to use in order to stabilize the system or to speed up response times. So there's a link somehow between natural living systems that have to work in the world and another kind of experiment, which is human engineering, which trains to use specific circuits again and again, amplifiers of a certain kind, operational amplifiers of a certain kind, memory registers of a certain kind. Except the processes are very different. Except when engineering is in early stages, it's like a tinkering process. When it's a mature field, you learn the circuits, a few circuits that can work in the real world. And this forms a link between a unified theory of things that need to function, whether they evolve or are engineered, which is a theory still to be made. We have the glimpses. And one of the glimpses is this concept of network motifs. We can learn from the biology what are the recurring circuits, and use considerations like what I did today with you to understand the different functions they might have. And there's additional functions we didn't talk about today. And we'll carry with this line of thought for another 1 or 2 lectures before switching to a new topic. So let's finish up. Let's take we can speed responses, stabilizes, evolution engineering. And last thing we'll do, if you want, feel like it, take one last nice sigh of relief. And thank you.